Good. Good morning, class. Chapter 5, Eukaryotic Cells and Microorganisms. Our learning outcomes will be to relate bacterial, archaeal, and eukaryotic cells to the last common ancestor. List the types of eukaryotic organisms and denote which are unicellular and which are multicellular. The first eukaryotic cells appeared on Earth 2 to 3 billion years ago. Bacteria and eukaryotes evolved from a precursor called the last common ancestor. They were neither prokaryotic nor eukaryotic. They gave rise to bacteria, archaea, and eukarya separately. The organelles originated from the primitive cells that became trapped in eukaryotic cells. Do it for me. Here is a picture of some ancient eukaryotic protists caught up in the fossilized rocks. Endosymbiosis. This talks about the larger precursor cell and the smaller prokaryotic cell. From single cell to multicellular, the first primitive eukaryotes were single cell and independent. They became specialized to perform a particular function in a colony. Complex multicellular organisms evolved when cells lost the ability to survive apart from the colony. And then we got tissues and organs. In microbiology, we're going to study eukaryotic organisms that are always unicellular, such as protozoa. Some that may be multicellular or unicellular, the fungi and the algae. And always multicellular are the helminths. Helminths have unicellular egg or larval forms. What is a helminth? Worms. So, concept check. Eukaryotes and prokaryotes evolved independently. True or false? All right. They came from a last common ancestor. What evolved from primitive cells that became trapped in larger cells? Describe the difference between tissues and organs. Now, you know this from previous biology classes. Tissues go to make up organs, okay? 3 types of eukaryotes that are studied. We're going to study the protozoans. Are we going to study fungi and algae and helminths? Here we're going to differentiate between cilia and flagella in eukaryotes. And I haven't looked in the last couple of days how many people have already done chapter 4 on Connect. Very good, very good. How many have not started it? Okay, you need to get started because next Thursday, we're having a quiz on chapters 4 and 5. We're going to describe the important characteristics of the glycocalyx in eukaryote, list which eukaryotic organisms have a cell wall because all of them don't, and list similarities between and differences between eukaryotic and bacterial cytoplasmic membranes. Some of the features of eukaryotic cells. These are found in all eukaryotic cells: the cytoplasmic membrane, the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton, and the glycocalyx. And in some eukaryotic cells, we have the cell wall, locomotor appendages, and chloroplasts. Here is a picture of the generalized eukaryotic cell. You can see there, what is this right here? That flagellum. What was the question on your Connect assignment about atrichus, monotrichus? What was that question, people who've done it? Say it again. I'm just going to look on the quiz. Do you like me looking on the quiz? The term that refers to the presence of flagella all over the cell surface is what? Peritrichous. Now, I might have this one on the quiz, but on the exam, I may ask the same question differently to where it would be the term that refers to the process of flagella on one end. And of these choices, peritrichous, monotrichous, amphitrichous, atrichous, what would that be? Mono. And then on the final exam, it may be different, but still it's the same question. So you need to know peritrichous, monotrichous, amphitrichous, all of them, okay? What else do we have here? We have the chloroplasts. Chloroplasts are involved in what process? Photosynthesis. Here's the centrioles. There is the cell wall. Here is the glycocalyx on the outside. It's the very outer covering. There's our smooth and rough endoplasmic reticulum. All of these, the nuclear membrane with pores, nucleus, nucleolus, are all within the nuclear envelope. Actin filaments, microtubules, intermediate filament, mitochondrion. What does the mitochondrion do? It's the powerhouse of the cell. Since eukaryotes have mitochondria, that's the powerhouse. But what about the other day we talked about prokaryotes? Do prokaryotes have a mitochondrion? So how do they get power? What generates the energy?
The cell wall.
The cell membrane. Membrane. Here are lysosomes and the Golgi apparatus. Flagella, eukaryotic versus bacterial flagella. What's the difference? This is 10 times thicker, structurally more complex. The eukaryotic is covered by an extension of the cell membrane. It has long, sheathed cylinder containing regularly spaced hollow microtubules, and it's in a 9+2 arrangement. Picture of the microtubules. Cilia is similar in overall structure to flagella, but it's shorter and more numerous, up to several thousand in some cells. It's found only in single-cell group of protozoa that certain animal cells. It functions as feeding and filtering structures on some cells. The glycocalyx. Remember, we looked at that pink glycocalyx on the outside of the prokaryote? Well, there's a glycocalyx here in the eukaryote. It's an outermost boundary that comes into direct contact with the environment. It's also called an extracellular matrix. It's composed of polysaccharides. It looks like a network of fibers, a slime layer, and a capsule. Then we have the cell wall that's found in fungi and algae. It's rigid and provides structural support and shape. Different chemical composition than bacterial cell walls. This one has chitin or cellulose in fungi. The cytoplasmic membrane, also known as just the cell membrane. It's a typical bilayer of phospholipids embedded with embedded protein molecules. It contains sterols. It stabilizes eukaryotic membranes. It's selectively permeable barriers. What does selectively permeable mean? Only certain things can pass through it. It's selectively permeable. Sophisticated mechanism for transporting nutrients and waste and other products. Describe 3 ways in which eukaryotic flagella are different than prokaryotic flagella. Name 1. Thicker, that is correct. What's the purpose of cilia? Filter? I'm going to go back a few slides here. Let's see, how do I go backwards? cilia. There it is. Its function as feeding and filtering structures on some cells. Okay, and we know some differences in prokaryotic and eukaryotic cell walls. Describe the main structural components of a nucleus. That's what we're going to talk about in the internal structures. Diagram how the nucleus, endoplasmic reticulum, and Golgi apparatus act together with vesicles during the transport process. We're going to explain the function of the mitochondrion, discuss the function of chloroplasts, explaining which cells contain them and how they arose, and we're going to explain the importance of ribosomes and differentiate between eukaryotic and bacterial types. List and describe 3 main fibers of the cytoskeleton. The nucleus, it's the control center. It's a compact sphere of and it is the most prominent organelle. The nucleus is separated from the cytoplasm by the what? The nuclear envelope. The nucleolus stains more intensely due to its RNA content, and it's the site of ribosomal RNA synthesis. RNA stands for, okay. Mitosis and meiosis, we all know what that is. How many people have taken 103? All right. Chromatin is the material of eukaryotic chromosomes. Units of genetic information are in the cell. Long linear DNA molecules are bound to histones, and it's visible during mitosis. Meiosis is the production of sex cells. So you can say that mitosis is the reproduction of non-sex cells, and meiosis is sex cells reproduction. The endoplasmic reticulum, there are 2 of them, the rough and the smooth, microscopic series of tunnels used in transport and storage. The rough endoplasmic reticulum has ribosomes attached to its surface. Proteins are held for packaging and transport. The smooth endoplasmic reticulum does not contain ribosomes. Its synthesis and storage of non-protein molecules is its function. Here is the origin and structure of the rough endoplasmic reticulum. You see all those little dots right there, those little blue dots? Those are the polyribosomes. Here is the nuclear envelope. These little openings are nucleopores. The Golgi apparatus is the site of protein modification and transport. It consists of a stack of flattened disc-shaped sacs closely connected to the endoplasmic reticulum. Where it is close to the Golgi apparatus, the ER forms transitional vesicles that are picked up by the Golgi apparatus. The Golgi apparatus adds polysaccharides and lipids to the proteins in the transitional vesicles, and it pinches off condensing vesicles. It conveys to lysosomes, or it's transported as secretory vesicles. So here's a picture of the Golgi apparatus. Does it look like a series of flattened sacs? Yes. Here are the condensing vesicles, those little circular endoplasmic reticulum. Okay. So the nucleus, the endoplasmic reticulum, and the Golgi apparatus are called nature's assembly line. Genetic information originates from the nucleus because what's stored in the nucleus? The chromosomes. And the chromosomes have what? Your DNA, your genes. Proteins are synthesized on ribosomes and deposited into the rough endoplasmic reticulum. Proteins are then transported to the Golgi apparatus to be chemically modified and packaged into vesicles. Here's the transport process right here. There is the Golgi apparatus. Yes. And it comes on out and goes into the Golgi apparatus, and there are some condensing vesicles, and we're going to see that it is going to continue into these vesicles, which are going to eventually end up in vacuoles. Lysosomes originate from the Golgi apparatus and contain a variety of enzymes. They are involved in intracellular digestion of food and protection against invading microorganisms. Vacuoles are membrane-bound sacs that contain fluids or solid particles to be digested, excreted, or stored, like the waste material. Mitochondria, we know it's the energy generator. It supplies the bulk of the energy of the cell. Its structure, it has a cristae and a matrix. It divides independently of the cell. It contains circular strands of DNA, and it has bacteria-sized 70S ribosomes. These characteristics provide evidence that mitochondria were cells engulfed by other cells and became their own organelles. Chloroplasts, what do chloroplasts look like to you? How do you describe them? Yeah, they look like the little chips, like a stack of them. Some say it looks like a stack of quarters or pancakes, okay? These are the chloroplasts. They are capable of converting the energy of sunlight into chemical energy through photosynthesis. Photosynthetic role of chloroplasts is they are the primary producers of all organic nutrients, and they produce oxygen. Ribosomes are protein synthesizers. Polyribosomes are short chain of ribosomes. And the full size of ribosomes is 80S, and that's a combination of the 60S and the 40S subunits. Here we have the cytoskeleton, and its function is to anchor the organelles, moving RNA in vesicles, permitting shape changes and movement. There are three main types of parts of the cytoskeleton: actin filaments, intermediate filaments, and microtubules. All right, major events of life in each organism type. We have genetics, reproduction, biosynthesis, respiration. Respiration occurs, the characteristic is the mitochondria. Photosynthesis involves the pigments and chloroplasts. Motility, locomotor structures. So what are the locomotor structures here? Flagella and cilia. What were the locomotor structures in the prokaryotic cells that we talked about the other day? Flagella was one. Another? Say it again. That is a type of protist itself. But just remember we talked about the fimbriae, the little hairs on the cell, and they help the cell to do what? Attach. What about, what else was the other one? Cilia, flagella? Now, there are three parts of the flagellum. It has three components. It's called the basal body, the hook, and the filament. That's one of your questions on chapter 4, remember? In your Connect. And where am I reading from right now? The quiz. So do you need to know that? The bacterium flagellum has how many parts, components? 3. The basal body, hook, and filament. Yeah, they have to be in order because it's asking you, like, what's the first thing? It's the basal body, and then the hook and the filament. The short, numerous appendages used by some bacterial cells for adhering to surfaces is called fimbriae. What about pili? Do you remember the word pili? Okay. The transfer of genes during bacterial conjugation involves rigid tubular appendages called sex pili. Okay? Just by way of Reminder. So now here in the eukaryotic cell, we only have the flagella and the cilia. Eukaryotic chromosomes are visible during all portions of the cell cycle? False. The blank endoplasmic reticulum is responsible for packaging and transport of proteins. Smooth, because the rough does what? What's on the rough endoplasmic reticulum? The ribosomes. What are vesicles that contain digestive enzymes? Mitochondria and chloroplasts contain 70S ribosomes and circular DNA. Is that true? Or false?
False.
Why do you say false? Again. Because it's 60-40? Okay, all right. And also, well, the DNA is circular. All right, so let's talk about the kingdom of fungi. Name me a fungus. Yeast, mold. What's another one? Huh? The ones that can be on your nails, and we hope we don't have any of that. Sometimes people have some toe jam. You know what toe jam is? Okay. It's just fungus. We don't want to have toe jam. We don't want to have any fungus on our toes. But we're going to learn the 3 features of fungal anatomy, differentiate between the terms heterotroph, saprobe, and parasite. Explain the relationship between fungal hyphae and the production of a mycelium. Describe 2 ways in which fungal spores arise and 2 detrimental and 2 beneficial activities of fungi from the viewpoint of humans. What's a common fungus that grows out in the yard or in a garden? Mushrooms. You like mushrooms? Like mushrooms. Don't like mushrooms. Some mushrooms are edible and they have good medicinal benefits. Some mushrooms are poisonous and should not be eaten. Kingdom fungi. Approximately 5 million species of fungi can be divided into 2 groups. The macroscopic fungi, such as mushrooms, puffballs, and gill fungi, and molds and yeast. Microscopic morphology. What does the word morphology mean? Shape. That's what it is. Shape. The yeast cell has a round to oval shape. It uses asexual reproduction. Hyphae are long thread-like cells found in filamentous fungi or molds, and pseudohyphae is chains of yeast. It can be dimorphic, which means that some fungi can take either form. Here's a picture of a fungal yeast cell. What do we use yeast for? What does it do? It makes it rise. Fungal nutrition. It's heterotrophic, which means it can't make its own, cannot. Autotrophic means it can make its own food. So fungus is heterotrophic. It acquires nutrients from a wide variety of substrates. Saprobes obtain substrates from dead plants and animals. Pay attention to this one because it's one of the questions. And parasites live on the bodies of living animals or plants. So what's the big difference in a saprobe and a parasite? Dead and living. So saprobes obtain substances from what? Dead plants and animals. And parasites live on the bodies of living animals or plants. Now, I wasn't really talking about this type toe jam. I was talking about the kind of toe jam that's usually right there under your little toe. But this is an example of substrates for fungi. See right there? And look at that person's rough heel there. Most microscopic fungi grow in loose associations or colonies. There's a mycelium, which is the woven intertwining mass of hyphae that makes up the body or the colony of a mold. Septa cross walls dividing hyphae into segments, and spores are fungal reproductive bodies. Using the rhizopus as an example, these are different types of hyphae, and rhizopus is a mold. So here is the vegetative hyphae and the reproductive hyphae, the spores there. Look at this piece of bread. What does it have? Mold. Mold rhizopus. Fungi have many complex and successful reproductive strategies. Simple outward growth of existing hyphae, fragmentation, a separated piece of mycelium can generate a whole new colony. The primary reproductive mode is spore formation. Fungal spores should not be confused with the more resistant non-reproductive bacterial endospores. So when we talk about spores related to fungus, we are not talking about bacterial endospores. What did we say the other day in chapter 4 about an endospore? Why? What is the purpose of a bacterial endospore? To protect it from harsh conditions. It could be a bacteria that loves high temperatures, but if it's placed in a very, very low temperature, the endospore helps protect it. Asexual spore formations, sporangiospores, are formed by successive cleavages within a sporangium. Conidiospores or conidia are free spores that are enclosed by a spore-bearing sac. Sexual spore formation has important variations occur when fungi of different genetic makeup combine their genetic material. Slight variations in the form and function are potentially advantageous to the adaptation and survival of the species. Identification in medical specimens, isolation on special media, observation microscopically and macroscopically. When we do our lab next week on microscopy, we're going to look at some prepared slides that have different types of fungus. We have the sexual spores that are difficult to detect, the asexual spore-forming structures used to identify genus and species, and we have a hyphal-type colony texture, pigmentation, physiological characteristics, and genetic makeup. Here are some representative fungi. All right. What are some medical conditions that are caused by fungi? Most human fungal infection occurs through accidental contact with soil, water, or dust. Pathogenic fungi, if it's pathogenic, that means it can cause disease. It can cause disease. Pathogenic fungi is a community-acquired infection that is caused by environmental pathogens. Hospital-associated infections caused by fungal pathogens in clinical settings. Did you know you could get an infection in the hospital? What's the most common one? That staph. On another name for staph, MRSA. Methicillin-resistant Staphylococcus aureus. MRSA. Staph. I had staph one time on my leg. It didn't come from the hospital. It came from being outside, picking up some leaves and trying to rake the yard for things that had been there all winter long. And as I was doing it, something just got on my leg and I started scratching it. And then I came in the house. And what should I have done first with that itch? Go take a shower. But I sat there and I scratched it. And I scratched it until I broke the skin. What happens when you break the skin? You let the infection on the inside. Staphylococcus aureus. It is a type of bacteria. Well, for me, it made my legs swell up, and then it went all over my body. Then I took 2 rounds of antibiotics, and it never would go away. So I started eating garlic every day. Yes, it makes your skin break out. You have the usual symptoms of fever, swelling. Yes. All right, other medical conditions caused by fungi are allergies and neurological conditions that are due to toxin reproduction. Major fungal infections of humans. On the outer epidermis, Tinea versicolor, and that's the causative fungus right there. Malassezia furfur. Say that. Malassezia furfur. You can have the epidermis of your hair, and the dermis can be attacked. Dermatophytosis, also called tinea or ringworm. You know what ringworm are, right? You've seen it. When I was small, ringworm was very common. Classmates used to have it on their face, ringworm in their hair, in their head. And it is caused by Microsporum trichophyton and epidermophyton. Then we can have something that affects the mucous membranes, the skin, and the nails. It can cause a yeast infection. We know what a yeast infection is. And the causative fungus is Candida albicans. Then we can have infections that affect the lungs and the skin. Other impacts of fungi, the negative industrial impact. The number of species are pathogenic to corn and grain. 40% of the fruit crop each year is consumed by fungi. It has a positive industrial impact. Its essential role in decomposing organic matter and returning essential minerals to the soil. They form stable associations with plant roots and increase water and nutrient absorption. Is that a good thing? Yes. Production of antibiotics, alcohol, organic acids, and vitamins. So fungus has a very good medical impact there. Food flavoring, alcohol in beer and wine, gas that causes bread to rise. Those are all impacts of fungi, positive ones. So the worst ones are the negative ones that are 40% of the fruit crop each year is consumed by fungi. That's bad. What is the type of fungus that derives nutrients from dead or decaying material? True, false. Fungal spores have a tough outer coating that allows them to survive in extreme conditions. True? You remember our going over that in this section? What are asexual spores contained in sac-like head, in a sac-like head? Pathogenic fungi require a host to complete their life cycle. Require a host? Okay, all right, that's false. The protists. We're going to look at some protists when we do the lab on the microbiology of pond water. let's see, we're supposed to be out at 9:45. Learning outcomes of the protists. We're going to note the protozoan characteristics that illustrate why they are informally placed into a single group. We're going to list 3 means of locomotion exhibited by protozoa, explain why a cyst stage may be useful, and give an example of a human disease caused by each of the 4 types of protozoa. So the protist is any eukaryotic, unicellular, or colonial organisms that lack true tissues. Within kingdom Protista, the protists, we have subkingdoms of algae and protozoa. The algae are photosynthetic protists. Have you heard on the news about the red tide that is happening right now in Tampa Bay? I told you already. Oh, okay. So if you stand there, right there by the bay, and smell it, it can cause nausea and other problems. And it kills the marine life, the fish, and the shellfish, and the shrimp, and whatever else is there. And it happens every year. It's just that this year, it is worse than it's been in a long time. The algae are a group of photosynthetic organisms where seaweed and kelp are the most recognizable. You ever been on a diet and taken something called kelp with lecithin? Never been on that diet, huh?
Yeah, I have.
The algae exhibit all of the eukaryotic organelles. Chloroplasts contain green chlorophyll. Other pigments create yellow, red, and brown coloration. The algae are widespread inhabitants of fresh and marine water. Plankton are floating communities of microscopic organisms. Their essential role in the aquatic food web. They play an essential role in the aquatic food web, and they produce most of the Earth's oxygen. Somewhere here in Alabama, they had some fish to come up dead, and they decided that it was because there was not enough oxygen in the water. So plankton produce most of the Earth's oxygen, so we need some floating community of microscopic organisms, don't we? Primary medical threat from algae is through ingestion of toxins during a what? Now that question is on one of your, when we do that laboratory exercise on pond water, that's one of the questions about red tide, okay? Biology of the protozoa. The protozoa include about 65,000 species. Most members are harmless inhabitants of the water and soil. A few species are parasites responsible for hundreds of millions of infections in humans per year. Protozoan form and function. Single cells containing all major eukaryotic organelles except chloroplasts. Organelles can be highly specialized into structures analogous to mouths, digestive systems, reproductive tracts, and legs, which would be a means of locomotion. Heterotrophic, they are heterotrophic and require their food in complex organic form. Free living species scavenge dead plant or animal debris, graze on live cells of bacteria and algae. The parasitic species lives on fluids of the host such as plasma and digestive juices, and they may actively feed on tissues. What is the, oh no, that's a bacteria. We're talking eukaryotic. I started to say that flesh-eating bacteria. Have you ever heard of it? Yes. Somebody got that down in Mobile Bay, Alabama this summer. It was on the news. And I want you all, I've already told you to listen to the news for anything that relates to microbiology. And if, what is the television show I want you to look at? the monsters inside. All right, what are the styles of locomotion? Pseudopods are what? False feet. They have an amoeboid motion. What does an amoeba do? Does it kind of glide along? It serves as a feeding structure. Flagella vary in the number from one to several, and cilia is distributed over the entire surface of the cell in characteristic patterns. Here are 4 types of locomotion. There is that flagellum right there. Here's a pseudopod. Look at all of these little extensions here. These are pseudopod, false feet that help it to glide along. Here, what is this right here? Cilia. And here we have a cytosome, food vacuoles, and the nucleus. Feeding in dormant stages. What does dormant mean? Like resting, like it's not active right now. It's dormant. A trophozoite is motile feeding stage. It requires ample food and moisture to remain active. A cyst is a dormant resting stage. It's formed when conditions become unfavorable for growth and feeding. It's an important factor in the spread of disease. Here's the general life cycle exhibited by many protozoans. It's active feeding stage of a trophozoite. Then the cell rounds up and loses motility. Then you have an early cyst, and then a mature cyst. And the cyst will break open, and then once it breaks open, the trophozoite is reactivated. Life cycles vary from simple to complex. Some protozoan groups exist only in a trophozoite state. Many types alternate between trophozoite and cyst stage, depending on the conditions of the habitat. Life cycle dictates the mode of transmission. So how do protozoans reproduce? They have simple asexual methods that usually they use mitosis. Several parasitic species reproduce by multiple fission. We have binary fission and we have multiple fission. Sexual reproduction also occurs. So is all protozoan reproduction asexual? No, you have sexual and asexual. Then we have conjugation. It's a form of genetic exchange between two cells. Those that use flagella to move and those that use amoeboid motion to move. The ones that use flagella alone, or flagella and amoeboid. They have a single nucleus. They sexually reproduce by syngamy. They form cysts and are free living. Those with an amoeboid motion to move are primarily amoeba. We could remember that. Amoeboid, amoeba. They use pseudopods for locomotion. Asexual reproduction by fission. They are mostly uninucleate, usually in cyst, and free living and not infectious. Those that use cilia to move, some have cilia in tufts for feeding and attachment. Most develop cysts. They have macronuclei and micronuclei. Division by transverse fission. Most have definite mouth and feeding organelle. They are free living and harmless. Those with no motility, sporozoa. Motility is absent in most of them. Complex life cycles with well-developed asexual and sexual stages, and the entire group is parasitic. Means it feeds on dead particles. True or false? False, because we know that if it's a parasite, it lives on something living. Most protozoa can be identified to the level of genus because of their unique appearance. Considerations in identification: shape and size of cell, type, number, and distribution of locomotor structures, presence of special organelles or cysts, number of nuclei, blood, sputum, cerebrospinal fluid, fecal or vaginal specimens are smeared on a slide with or without staining. Some important protozoan pathogens. Pathogens cause disease. Parasitology is the study of protozoa and helminths. A parasite is a term that's most often used to denote protozoan and helminth pathogens. Some other important protozoan pathogens are Giardia lamblia. It causes intestinal distress. Trichomonas vaginalis. It causes trichomonasis. Have you heard of that before? It's in humans, caused by the protozoa. Trichomonas vaginalis. And what did you say? What does it cause? Vaginal symptoms. Kind of like a yeast infection. You know what that is? Okay. Leishmania donovani. Ooh. It causes leishmaniasis, either skin lesions or widespread involvement of internal organs. Its reservoir is in animals and vector-borne. Then you have some non-motile protozoa that also cause disease. Plasmodium vivax causes malaria. Toxoplasma gondii causes toxoplasmosis. Cryptosporidium causes cryptosporidiosis. And Cyclospora cayetanensis causes intestinal and other symptoms with cyclosporiasis. Trypanosome is a category for pathogenic flagellates. Trypanosoma brucea causes African sleeping sickness. Trypanosoma cruzi causes Chagas disease. Long crescent-shaped cells with a single flagellum. They are both found in the blood during infection, and both can be transmitted by blood-sucking vectors. Algae don't cause any type of human disease. Everyone, it's caused by trypanosoma. Say it right, Janice. Trypanosoma brucea. I'm not sure. Somebody on your phone right now, look up Trypanosoma. Who's got a phone? Trypanosoma. Brucei. Brucei. Anybody can get African sleeping sickness if you have this particular pathogenic flagella in your system. Anybody got it? Okay, what about sub-Saharan Africa?
It says the carabao that flies in that area.
Okay, carabao flies in the area. Okay. So if we had some of those flies here, then you might get bit by it, huh? Name me 3 styles of protozoan locomotion. They can have, what do we call pseudo? Name me 3 types of false feet, okay? Pseudopods. What's another kind of locomotion? Okay, what's another one? All right, it says the term what is most often used to describe protozoan and helminth pathogens. Cilia, we didn't name cilia. Pseudopod, cilia, and flagellum. All right. Go. So next we're going to learn about the parasitic helminths. We will pick up on Monday with section 5.6 on the helminths. We're going to get ready to go to the lab. What are we doing in lab today? We are having our first peer review of our lab notebook today, so we get started on the right path. And then when are we doing microscopy? Next week, Tuesday. You like that? All right, now I'm going to pick up with 5.6.